Hi everybody. In this class we are going to discuss about the meiosis process. Okay, meiosis process. In the previous class we discussed about mitosis. So here we are going to discuss about meiosis. So meiosis is a very important process in the higher animals which are undergoing sexual reproduction. Okay, which are undergoing mainly sexual reproduction. But of course it is also important in the lower organisms also. But here we are going to understand how this meiosis process is taking place. So when we say that in sexual reproduction meiosis is very important, how? Okay, how this meiosis process is important. So let us learn about this. So during sexual reproduction, fusion of male gamete and female gamete will be taking place. Okay, there will be a male gamete and there will be a female gamete. Okay, these two will join together to form as what? The offspring. Okay, we are calling it as the offspring. So this is common process in what? Sexual reproduction. So here the basic point that we have to remember that the gametes are involved in the fusion. Okay, the gametes are involved in the process of sexual reproduction where they fuse together. Okay, they fuse together to form as what? Offspring. Otherwise we call this with also another name called as what? Zygote. Okay, we call it as the zygote, of course. Okay, zygote. Now here please remember what is fusing male gamete and female gamete. Now the question is, what does this male gamete and female gamete contains? Okay, what does this male gamete and female gamete contains? The male gamete and the female gamete will be carrying what? Chromosomes. Okay, male gamete and the female gamete are carrying the chromosomes. Please remember this. Okay, male gamete and the female gamete will be carrying chromosomes. As we all know that every cell in the human beings, okay, every cell in the human beings, let it be any cell from any part of our body, okay, any cell from any part of our body will be consisting of how many chromosomes? 46 chromosomes. Okay, it consists of 46 chromosomes which are present as how? As pairs. Okay, they are present as pairs. So when these 46 chromosomes are paired, we call them as what? There are 23 pairs of chromosomes in each cell. Please remember, this is in each cell. Okay, so let us remember that this is in each cell. Okay, in each cell of our body. Okay, in each cell of our body. Please remember that. Now here when we say that there are 23 pairs of chromosomes or otherwise as 46 chromosomes, how are they carried? How are they carried? Remember that the male gamete and the female gamete will be carrying the chromosomes only half of the number of this. Only half of the number of these chromosomes will be carried by what? Male gamete and the female gamete. So this is a very important point. Even though we say that each cell is containing how many chromosomes? 46 chromosomes, or otherwise when these 46 are paired, we are calling them as 23 pairs. But remember that a male gamete and the female gamete will be carrying only half of their number, which means here if you are taking the male gamete, it will be carrying how many chromosomes? Only 23 chromosomes, not 23 pairs, nor we cannot call them as 46. So only 23 chromosomes. And in the same way here, the female gamete is also carrying how many chromosomes? Only 23 chromosomes. Okay, only 23 chromosomes. So here, this is what is very important. Okay, so remember that whenever a cell is having the full set of chromosomes, okay, full set means total 46 chromosomes, we use one word called as diploid. Okay, we use one word called as diploid. So here we can represent it as 2n. N means what? Number of chromosomes. Okay, 2n. N means number of chromosomes. So here 2 into 23, that gives rise to how many chromosomes? 46 chromosomes. So whenever it is 2n, we should remember it is diploid. Every cell in our body is diploid. Every cell in our body is diploid, except what? The male gametes and the female gametes. Okay, male gametes and the female gametes. Here the male gamete, okay, the male gamete and the female gamete which are carrying half of the number of chromosomes will be called as what? Haploid. So male gamete is what now? Haploid. The female gamete is called as what? Haploid. Okay, so here we have to remember that male gamete and female gametes are always haploid. Haploid means what? 1n we say. Okay, 1n means what? Only they will have what? 23 chromosomes only. They will have only 23 chromosomes, not 46. They will not carry 46 number of chromosomes. What happened here? The number of chromosomes reduced to half. The number of chromosomes reduced to half. That's what we have to remember. So we have to remember this word now, haploid. And another word here is diploid. Okay, haploid and diploid we have to remember. So that's why we are saying gametes. What are those? Male gametes and female gametes are always what? They are haploid. Okay, they are haploid. Haploid means only one end we are saying. One end means it is carrying only 23 chromosomes. Okay, only 23 chromosomes are carried. So always remember gametes are haploid. Okay, but all the other body cells are what? They are diploid. Okay, they are diploid. Remember that. Now gametes are formed from specialized diploid cells. These cells called as gamete cells, male gamete and the female gamete, are formed from the diploid cell. Which means if you are taking a diploid cell which is having 23 pairs, or otherwise we can also say that this is having how many chromosomes? 46 chromosomes. Okay, 46. This is a specialized diploid cell. Okay, because it has a full set of chromosomes, not half the set. So when this having full set as 23 pairs, otherwise 46. Okay, this cell will divide and give rise to here only 23 chromosomes. Here also 23 chromosomes. So this process, if it is taking place in the males, we call these cells as what? Male gametes. Okay, we call these cells as male gametes. If it is taking place in the females, we call them as female gametes. But otherwise, same thing, you can also just, we will be keeping like this, okay? Actually, here like this, which means from one specialized diploid cell, how many cells are formed here now? There are 4 cells that will be formed. 4 cells are formed from one specialized diploid cell. All these 4 cells are carrying half of the number of chromosomes. Half of the number of chromosomes. That's the point we have to remember when we talk about the third point. That is, gametes are formed from specialized diploid cells. Okay, next point. In this process, the number of chromosomes will be reduced to half. So what happened in this process now? The number of chromosomes reduced to half. When there are 46 chromosomes, half of the 46 is 23. So they reduce to half. So this process in which the number of chromosomes will be reduced to half will be called as what? Meiosis. Okay, will be called as meiosis. So what is meiosis now? When the number of chromosomes will be reduced to half, we call it as meiosis. Okay, we call it as meiosis. And note, we have to remember these 2 points or 3 points. Okay, very important points. Meiosis ensures that the production of haploid phase in the life cycle of an organism. In the life cycle of an organism, meiosis is ensuring that there will be production of what? Always the haploid cells. Okay, always the haploid cells. Meiosis is taking it for granted that, okay, it is guaranteeing, okay, it is guaranteeing that there will be production of what? Half of the cells, or half of the chromosomes in each cell, that which we are calling them as male gametes or female gametes. Okay, so that's the point we have to remember. And once meiosis is reducing the number of chromosomes, then here these male gamete and the female gamete which fused together to form as the offspring, otherwise called as what? Zygote, will be having what now? It is having 2n. Means what? 2n means what? Diploid cell. Diploid cell means what? It is having again 23 pairs of chromosomes. Otherwise we call it as 46 chromosomes. Okay, 46 chromosomes. So here by what process it is restoring the number of chromosomes? It is by a process called as what? The fertilization. So fertilization restores the diploid phase. Okay, meiosis is reducing the number of chromosomes into half, whereas the fertilization is again restoring the diploid condition in zygote. So meiosis, remember, occurs in both plants and animals. Okay, meiosis is taking place in both plants and animals in or during gametogenesis. Okay, during gametogenesis. What do you mean by gametogenesis? The process of formation of gametes. If it is in males, male gametes we say. If it is in females, we call them as female gametes. We call them as female gametes. We will see some key features. Okay, we will see some key features of the meiosis. First feature is that in meiosis we will be seeing two sequential cycles. We will be seeing two sequential cycles. One is called as what? Nuclear division, and the other one is called as cell division. Okay. So in meiosis, they will be seeing nucleus will be dividing, and after the nucleus division, what happens? Even the cell also will be dividing. That's what we said in the previous class as what? The karyokinesis. Okay. We said it as karyokinesis. Okay. Karyokinesis. And other one we said it as what? Cytokinesis. Okay. Cytokinesis. So these two events will be taking place in meiosis, which consists of, or these two events are called as what? Meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. So meiosis 1 has these phases, as again it is subdivided as what? Prophase 1, metaphase 1, anaphase 1, telophase 1. And meiosis 2, again it is subdivided as prophase 2, metaphase 2, anaphase 2, telophase 2. These are the subdivisions, okay, of meiosis 1 and meiosis 2. So that is the first feature that we have to remember when we talk about the meiosis. And meiosis involves pairing of homologous chromosomes. Here the word homologous means what? Identical chromosomes. Okay, identical chromosomes will come and join with each other as a pair. And they exchange the genetic material so that they will be resembling the same. They will be resembling the same in all the characteristics or in all the genes. Genes present on one chromosome will also be present on the other chromosome. So that's why they will be pairing. Okay. And the next one is at the end of meiosis. Once the meiosis is ended by cytokinesis, there will be 4 haploid cells that are formed. It's a very important point. Okay. 4 haploid cells will be formed at the end of meiosis. At the end of mitosis, only 2 cells are formed which are diploid. Here haploid cells are formed at the end of meiosis 2. Okay, at the end of meiosis 2. So these are the points we should remember them as what? Key features. Okay, key features. So now we will see the first phase of meiosis 1. Okay, meiosis 1, prophase 1. Remember, whenever you are discussing about this cell division, you should remember this question that prophase 1 is the longest phase. Okay, prophase 1 is the longest phase where it is again subdivided. Okay, it is again subdivided as what? The leptotin, zygotin, achatin, diplotin and diakinesis. All these stages can be visible or seen under the prophase 1. So prophase 1 is the longest phase. Okay, prophase 1 is the longest phase. And first one, if you are seeing the leptotin, here in leptotin stage, the number of the chromatin network, okay, means the chromatin network that is there in this nucleus. Okay, let me say that this is the nucleus. Okay, this is the nucleus, and let us say that this is the cell membrane. Okay, this is the cell membrane. So here, when you look at the nucleus, it will be very clearly visible, distinct. Okay, it is very clearly visible under the electronic microscope, and here you can see very clearly even the chromatin network also. Okay, you can see the chromatin network also very clearly. Okay, chromatin network also can be seen very clearly. Okay, chromatin network, and this is more, that's why we say that in leptotin stage the chromatin network is very visible and compact. Okay, very thick, it is very closely present. Okay, it is very thick, so we can say that it is appearing very thick. Okay, when we see it under the microscope, and that's why we say that now this cell is ready to undergo meiosis. Okay, ready to undergo meiosis. Now that is in the leptotin stage. And zygotin, during if you are now in the leptotin stage only, we will see that this is the chromatin network we are seeing. In the leptotin stage, this chromatin network will again separate as individual chromosomes. We said that this cell is a specialized diploid cell. Okay, this cell is what? It is a specialized diploid cell. Okay, it is a specialized diploid cell. Okay, specialized diploid cell. Now this specialized diploid cell is having the chromatin network in a very visible and in a compact form, and this chromatin network now separates. It separates into individual chromosomes. It separates into individual chromosomes. Means how many chromosomes will be there here now? There will be 46 chromosomes that will be present. Okay, this is a diploid cell we are saying. 46 chromosomes will be there. Otherwise, how many pairs it can be? It can be 23 pairs. Okay, it can be 23 pairs. Now here, once they separate into individual chromosomes, these chromosomes, each chromosome now separates, okay, divides longitudinally and forms as sister chromatids which means if this is a chromosome, okay, this is the chromosome. Each chromosome divides longitudinally but held at one point called as the centromere. Okay, and when it divides, now how it appears? It is appearing like this. Okay, it is appearing like this as though it is held at one point. So here we are seeing one chromosome having two strands or chromatids we call, because these two arms, okay, these two arms are arising from the same centromere. We call them as what now? We can call them as sister chromatids. Okay, we call them as sister chromatids. So what are sister chromatids means? When the arms of the chromosome are attached to the same point, that is centromere, we call them as sister chromatids. So every chromosome, how many chromosomes? 46 chromosomes. Every chromosome will be appearing like this now. Okay, will be appearing like this. After which, okay, now, okay, after which what happens is that all the chromosomes will be aligned. Okay, homologous chromosomes means these two are identical among those 23 pairs or otherwise 46. Two chromosomes are identical exactly. So those two which are identical will be pairing. Okay, so we have to remember that here the homologous chromosomes are pairing. Okay, homologous. Okay, pairing of homologous chromosomes. Okay, homologous chromosomes, and this process, okay, pairing of homologous chromosomes can also be called as what? Okay, can also be called as what? Synapses. Okay, we call them as synapses. So that we are seeing here in the zygotin stage. Zygotin. So here in zygotin, this is called as what now? Zygotin. Okay, zygotin. Please remember this. Okay, all these three stages are showing the first stage called as leptotin. So the zygotin is showing what? Pairing of homologous chromosomes. Pairing of homologous chromosomes is also called as synapses. Please remember this word, a very important word. Okay, very important word, synapses. Pairing of homologous chromosomes. So when the chromosomes are paired, okay, identical chromosomes, two chromosomes which are, okay, has pairs, okay, which are as pairs. Okay, which are as pairs there. Okay, two identical chromosomes which are as pairs, we are calling it as synapses. Now once they are as pairs, okay, how many pairs will be there now here? 23 pairs will be there. 23 pairs of chromosomes are there. But each chromosome is having two arms, and these arms are called as what? Sister chromatids. Okay, sister chromatids. Now when we are using here two words. Okay, bivalent or tetrad. What is this bivalent or tetrad? Okay, bivalent or tetrad. See here, when it is there, okay, as two chromosomes as pairs, we use the word bivalent. Okay, we use the word bivalent. In the bivalent, we are seeing how many arms? 1, 2, 3, 4 are there. So that's why we are calling it as tetrad. Okay, that's why we are calling it as tetrad. Next one is diplotin. Okay, next one is diplotin. It starts from separating from each other. Okay, separating from each other. Now if you are seeing here, I am showing you here in the diagram only one pair of chromosomes, homologous chromosomes, which have undergone the synapsis process. Okay, which have undergone the synapsis process. Synapsis means what? Pairing of homologous chromosomes. So pairing will be seen in all the 23 chromosomes. 23 chromosomes as pairs. So that's why we are calling it as 23 pairs. Means you can also say that pairing of chromosomes, homologous chromosomes, is taking place in all the 46. So once they pair, they will be as 23 pairs. So like this, there will be 23 pairs. I am taking only one for us to understand in this topic. But what we are going to learn from this one pair is applied for all the other chromosomes. Okay, 22 pairs also. So here if you are seeing, there is pairing here. One arm of one chromosome, okay, here if you are seeing, is coming and joining with the another arm here at this point. Okay, this point. So see here now, this is one arm of the chromosome, this is another arm of the chromosome. This one arm is coming and it is crossing with the other arm of other chromosome here, both. Okay, this arm is up from this chromosome, this arm is from this chromosome. So when they cross with each other, they will be appearing here. If you are seeing clearly, it is appearing like what? X shape. It is appearing like what? X shape. Please remember this. Okay, it is appearing like X shape here. If you are looking at here, here also it is appearing like X shape. So here X shaped part where there is crossing over, we are calling it as what? Chiasmata. Okay, we are calling it as chiasmata. Okay, please remember it as chiasmata. Please remember that. So the point at where the crossing over occurs, okay, the point at where the crossing over occurs and this point where it crosses is called as what? Chiasmata. Okay, chiasmata we are calling it as. So chiasmata occurs and that is in the diplotin stage. Remember one point and a very important point. Very important point is that in vertebrates, those which are having the backbone, their oocytes, okay, their oocytes will be arrested at this stage for few months or years. Means the oocytes will be remaining in diplotin stage for few months or years. Later they will be undergoing the other process called as diakinesis. So now the next one is diakinesis. Diakinesis means what now? Now they slowly separate from each other. Now if you see, these two are very close, but here the same chromosomes, they are, say, suppose if these are the chromosomes, okay, here they are like this and they are now slowly they are getting separated. Okay, so that separation is called as what? We are calling it as diakinesis. This separation, okay, diakinesis will be taking place completely, this separation of the homologous chromosomes will be taking place completely in the diakinesis. It will be starting in diplotin. Separation of the chromosomes will be taking place in the starting in the diplotin and it ends up in the diakinesis. It ends up in the diakinesis and they will completely get separated. Now the crucial point is that you have to remember where they have crossed. Okay, there they have undergone what? Crossing over. This is the point. Okay, they have undergone crossing over in packaging stage, this area. Crossing over means what? It is the exchange of genetic material. Exchange of genetic material. Because there is exchange of genetic material in the packaging stage, both the chromosomes, once they get separated, they will be identical, exact copies. The gene which is there present in one chromosome will also be there present in the other chromosome. So that's what is the main point we have to remember when we talk about the prophase 1. So that crossing is taking place, crossing over is taking place in packaging stage. And for crossing over, remember it requires one enzyme called as recombinase. Okay, it requires one enzyme called as recombinase. So once they exchange the genetic material, by the end of diakinesis, both the chromosomes will get separated. So this process will be taking place in all the 23 pairs of chromosomes, and all the 23 pairs of chromosomes are now having the genetic material which is exchanged with each other during prophase 1. Okay, during prophase 1. And the next one is metaphase 1. Okay, metaphase 1. Okay, under meiosis 1, metaphase 1. So what happens during the metaphase 1? Bivalent chromosomes, okay, bivalent chromosomes align in the equatorial plate. Means this is the equatorial plate we say. In this diagram, if you are looking at, this area is called as what? The equatorial plate. And we are using the word bivalent, okay? We are using the word bivalent. Bivalent means what? Here, this is one chromosome, okay, which we studied that it is separated into two, okay, arms, and here is one, okay, here is another arm, and if you are taking the same way another chromosome, okay, say suppose this is the one which is separated at the center and it is forming as the two arms like this, okay, two arms held at one point, okay, here are the two chromosomes. So when you are talking first chromosome, second chromosome, these two are called as what? We are calling them as what? The bivalent, okay, bivalent. But otherwise, if you are calculating the number of arms, first arm, second arm, third, fourth, we are using what? Tetrad, okay, we are using the word tetrad. Please remember that, okay, tetrad. So here we are saying that microtubules Microtubules are nothing but they are proteinaceous material, okay, proteinaceous material, okay, attached to the pair of homologous chromosomes. Now here are the chromosomes. So these are called as what now? The microtubules, okay, microtubules, okay, these are called as the microtubules. Otherwise, we can also call them as what? Spindle fibers, okay, we can also call them as spindle fibers, okay, we can also call them as spindle fibers. They are made up of proteins, okay, they are made up of proteins. So from the opposite poles, these microtubules will come and attach to the chromosomes, okay, attach to the homologous chromosomes. Clearly you have to see now, okay, we said that at the center all the 23 pairs of chromosomes are there present. It is not 1, 2, 3, 4. Only in diagram we are taking like that. But otherwise at the center called as the equatorial plate, how many chromosomes will be there? 23 pairs. This is first pair, second pair, third, fourth, fifth, sixth, seventh, eighth, ninth, so on. And 23 pairs will be there at the center, which we are calling it as the equatorial plate. And if you are clearly seeing here, spindle fiber, or otherwise called as microtubule, is formed from one pole, is coming and attaching to one chromosome only, which is having two chromatids. Remember that. This microtubule is coming and attaching to this only, okay, which is having two chromatids. That's the key point we have to remember. So that is about what now? The metaphase, okay, metaphase. So how to identify that the chromosomes are there in metaphase stage? They will be there at the center of the cell, which we are calling as equatorial plate. And the next one is anaphase 1. Anaphase 1. Homologous chromosomes separate. Okay, homologous chromosomes separate. Okay, homologous chromosomes are separating where the sister chromatids remain attached to the centromere. So if you are seeing here, these are the sister chromatids, we can say. Okay, these are the sister chromatids. Okay, we can say like this. Okay, having both the arms. Okay, having both the arms like this. Okay, having both the arms. Okay, both the arms are there. Sister chromatids. So here this is pulled towards the opposite pole and this is pulled towards the opposite pole. This is happening during the anaphase. Okay, so they are pulled towards the opposite poles. Okay, they are pulled towards the opposite poles. So that's what we are saying during the anaphase. Homologous chromosomes separate. Identical chromosomes. These two we said what? Identical chromosomes. That's what we have seen here. These two are identical chromosomes. They are separating from each other. But still, what we are saying, sister chromatids remain attached to centromere. Sister chromatids means what? See, again I am showing you. These are, okay, these two are what? Sister chromatids arising from the same centromere. That's what we have to remember when we talk about the anaphase 1. Anaphase 1. Now, the next one is telophase 1. Okay, telophase 1. Here in telophase 1, the nuclear membrane and nucleolus that disappeared during which phase? Prophase 1 will be reappearing during the telophase 1. It is reappearing. Okay, it is reappearing. So here, if this is the cell, okay, if this is the cell, now slowly there will be formation of what? The nuclear membrane and also the nucleolus. Okay, and also the nucleolus. So here let us say that this is the nuclear membrane. Okay, this is the nuclear membrane and here is the nucleolus. Okay, here is the nucleolus. Okay, nucleolus. And now what else is seen in the telophase 1? We should remember that here in this cell the spindle fibers, which we call them as what? Microtubules, are bringing the chromatids like this. Okay, the chromosomes. Okay, chromatids or chromosomes will be brought. How many chromosomes will be there at each pole? Okay, how many chromosomes are there at each pole? So here they are 23. Okay, here also there will be 23. And now once they are there here as 23 and here as 23, now what happens is this cell, okay, now undergoes this cytokinesis. Cytokinesis means what? Cell division. The cell divides. The cell divides into two cells. Here is one cell and here is one cell. Okay, two cells are formed. Two cells are formed. Please remember that we are calling it as what? Cytokinesis, division of cytoplasm of the cell. So here how many chromosomes will be there? 23 chromosomes. Here also how many chromosomes are there? 23 chromosomes, which will be appearing as the chromatin network. Okay, it will be appearing as the chromatin network. So there are two cells formed. But what we said, at the end of meiosis, there will be 4 cells that will be formed. All these 4 cells are haploid. All these 4 cells are haploid. So after the end of cytokinesis, please remember, this is all about meiosis 1. Okay, this is all about meiosis 1. So right after the meiosis 1, these 2 cells will be taking a short-lived, okay, a period of rest, which we call it as interkinesis. Okay, which we call it as interkinesis. Okay, which we call it as interkinesis. Okay, and then these 2 cells will be entering into meiosis 2. Meiosis 2. So now we will discuss about meiosis 2. Okay, we will discuss about meiosis 2. Meiosis 2 is exactly the same as mitosis. Okay, meiosis 2 is exactly the same as mitosis. So we will see only in diagrammatic form because we already understood mitosis. Okay, so now here, as we already just completed that, right after the meiosis 1, there are 2 cells which are now, here are those 2 cells having 23 chromosomes, here having 23 chromosomes. Now starts with meiosis 2. Meiosis 2. So meiosis 2 is also having what? Prophase 2. In prophase 2, what happens? The same. The chromatin network separates it as individual chromosomes. Each individual chromosome, okay, if this is the individual chromosome, it has also dividing longitudinally, held at one point, and having how many arms now? It is also having 2 arms now, held at one point. So all those chromosomes are there here now in both the cells. Okay, so each chromosome is having what now? The chromatids. Okay, each chromosome is having the chromatids. So, okay, they are all there present. And in prophase 2, by the end of prophase 2, we are seeing the chromatids and the nuclear membrane disappears. Nuclear membrane disappears and all the chromosomes with their chromatids are scattered in the cytoplasm of the cell. That is in the prophase 2. Metaphase 2. In metaphase 2, what happens? The microtubules from the opposite poles come and attach to the chromatids, okay, of the chromosomes. Okay, they will be attached to the chromosomes, which means if this is the, okay, if this is 1, okay, just follow this. If this is 1 and here is the other one, they will be separated. The 2 chromatids, okay, the 2 chromatids. This is one chromatid and this is another chromatid. The 2 chromatids will separate, which means here they are going up with the microtubule. Here is another one, okay, going down with the pulled by the microtubule. Okay, so here they are held by the microtubules, and in the metaphase 2 we know that it is all the chromosomes will be present at the center, and it is called as what? Equatorial plate. The spindle fibers are attached. That is in the metaphase 2. Anaphase 2, what happens? This is what? They get separated from each other. Okay, the chromatids are getting separated from each other, but in the meiosis 1, the chromatids will not separate. Okay, they are held and they are pulled. But here the chromatids are separated and each chromatid is pulled towards the opposite poles. Okay, this is in the anaphase 2. So by the end of anaphase 2, the chromatids will be pulled towards the opposite poles. And after reaching the opposite poles, again all the chromatids, say suppose this is 1, 2, 3, 4, 5, here 1, 2, 3, 4, 5, 6, like that, 23 here, 23 here are there. Okay, in anaphase 2, I am talking about this anaphase 2. All these will be forming as a network, which we call them as what? Chromatin network. Okay, they will form as again the chromatin network. Enclosed by what now? The nuclear membrane. Please remember, in prophase 2, the nuclear membrane disappeared, we said. The nuclear membrane that disappeared during the prophase 2 will be again reappearing during the telophase 2, forming as the chromatin network. So once the chromatin network is formed, okay, by the end of telophase 2, there will be nuclear membrane that reappears. Now there are 2 nucleus in each cell. Here also the same. Okay, and now these 2 cells ending up telophase 2 undergo what now? Cytokinesis. Now till now we have seen division of nucleus, which we call it as what? Karyokinesis. Okay, karyokinesis. Now what is happening is the 2 of this cell divides by undergoing what process now? Cytokinesis. Cytokinesis. Now once they divide, there are 2 cells. Each cell is having 23 chromosomes. Here is 23 chromosomes. Here. So from one cell there are two cells. From one cell, two cells. But otherwise we are saying how many cells are there now present here? There are 4 cells that are present at the end of meiosis. And all these 4 cells are what? They are haploid. Okay, they are haploid. So this process is taking place both in the plant cells and animal cells, only in the specialized diploid cells, which will be giving rise to what? The male gametes. Okay, which will be giving rise to the male gametes, and they will be giving rise to the female gametes if it is taking place in the females. Okay, so if it is taking place in the males, these are called as male gametes. If the process is taking place in the females, it is called as female gametes. So this is all about the meiosis. So we have to remember that in meiosis it is a division where 4 cells are formed, and because of that, okay, these 4 cells are haploid. Okay, 4 cells are formed. Those 4 cells are haploid, and which means the number of chromosomes are reduced to half. That's why meiosis is also called as what? Reductional division. Please remember this. Reductional division. Please remember this point. Okay, reductional division. So these are the points which we should keep in mind when we are talking about the review of mitosis, which we studied previously, and the review of meiosis. Thank you so much.